welcome to the video looking at the structure of plasma membranes. I was specifically looking at the fluid mosaic model, which is now what is used to describe plasma membranes. If we look at why it's actually called the fluid mosaic model, this is because the membrane consists of a phospholipid bilayer. We'll go on to that in a minute, but we have proteins that are floating in the membrane, so we've got a flexible movement. Proteins, lipid molecules and things can be attached to each other or free moving, so we've got quite a lot of flexibility in the movement of the membrane. So if we're looking at the different components, then we can see how it's quite like a mosaic model because we've got lots of different components in there. We've got the many different kinds of proteins here, which I've represented in the colour green. So we've got a channel protein and a carrier protein, and these are both used for facilitated diffusion and some for active transport, but we will go on to that in another video in more detail. We've also got peripheral proteins that are just on the outside of the membrane, and they can also be called extrinsic proteins. We've got receptor sites that are protein receptors as well. We've got the kind of carbohydrates molecules that are in like a purple, and in pink we've got the phospholipids here. And what we can see here, we've got the phospholipid at the head, and then we've got the tails. So we've got these fatty acid tails, and these are hydrophobic, and that's why they face inwards on the membrane. So they repel water, hydrophobic, but then the phosphate head is hydrophilic, and they face outwards, and then so we've got a double layer here like that. And this is why some molecules cannot pass through the membrane by diffusion, because of the hydrophobic polar regions. So if we're looking at the cholesterol, for example, that I've put here, cholesterol, its role is just to give mechanical stability and flexibility. There are two rules for the cholesterol within the membrane. And then we have the glycolipid and glycoproteins. We can see that they're quite similar, but this carbohydrate region is attached to a lipid, whereas this is attached to another protein, such as why it's a glycoprotein or a glycolipid. Glycoproteins then, they have quite diverse functions. They can have roles in the immune system, so immunology, with white blood cells. They attach to the blood vessel lining the glycoprotein called lectins. So without this, the white blood cells won't be able to travel throughout the body. So, and we've also got the glycoproteins are important for protection, so involved in keeping skin healthy on the surface of the skin, like the epithelial cells, which help attach skin cells to each other, forming a tough barrier to protect the body. So we've got protection and immune responses, really. For the glycolipids, these are important for cell-to-cell interaction, so cell signalling. So like a recognition site, so the saccharide of the glycolipid will bind to a specific complementary carbohydrate or to a lectin, which is a carbohydrate binding protein, and this is the basis of the cellular responses, which contribute to activities such as regulation and growth. So again, we've got growth and regulation are just two examples. Again, just looking at cholesterol, we've said about flexibility, but it does also regulates the membrane fluidity. So flexibility, and we can also say that fluidity is what it's kind of regulating the fluid part of the fluid mosaic model. Cholesterol is also a part of a signalling molecule, so in the myelin sheath, which we've talked about in the nerves topic, the myelin sheath is rich in cholesterol because it provides protection. These peripheral proteins, they're entirely on the outside, like we've said, and they're bound by weak intermolecular attractions such as the hydrogen bonds. I'm not going to go too much into channel proteins and carrier proteins here. I will be going into more detail about those in the other videos. And I think that is all for this video. Thank you for watching.